Please join me in welcoming to the Distinctive Voices podium, Dr. Katiem Paul.
Thank you so much. Thank you for that great introduction. So today I'm going to talk to you about how genes determine our quality of sleep, but I'm going to tell you the story first about giving you a background about some of our more surprising discoveries in the field about the genetics behind sleep, and then I'm going to talk to you about research in my lab, research that has been published, and I'm actually going to, hopefully if I have time, to show you some of our newer discoveries in our lab. So I study sleep. And why? Because sleep is essential to health. We spend, or we should be spending, about a third of our lives in this condition that we call sleep. We spend so much time in it, it's obviously important, so much so that if you don't do it, it is harmful to your health. Sleep is very well conserved across phylogeny, across the myriad different species, animal species. So something that consumes such a large chunk of your life and is so well conserved must be driven by a tractable set of genes and genetic and molecular factors. And that's what I do for a living, is I try to identify the genes and the genetic networks that are responsible, that help drive and help encode this very important process. And that can be difficult to do for several different reasons. It has been a challenge. One of the larger overarching reasons why it can be difficult to try to identify the genes and gene networks that encode sleep is because sleep is not the easiest thing to define. It's not the easiest thing to really kind of put in a box. For each one of us, if we were asked to define our own sleep, probably wouldn't be very difficult. Something like, nighttime comes, you finish watching television or scrolling on the computer, you get in bed, you have some sleep latency until you fall asleep. If you snore, you may have a couple of brief awakenings that you may not be aware of. If your bed partner snores, you'll probably have some brief awakenings that you will be aware of, and they may not be very pleasant. And sometimes you'll be wakened by an alarm clock, which you may hit snooze on several times before you actually get out of the bed. Not everyone sleeps like that. And if you consider sleep across the myriad species, not just humans, mammalian species and non-mammalian species that sleep in different environments, sleep in the water, sleep on land, sleep high perched on trees and so forth, then you can appreciate that sleep can be a bit more difficult to define. And if it's not easy to define, it presents challenges to try to understand the genetic regulation of. I've seen a lot of different definitions of sleep. This is my favorite definition because of the economy of words. I got this definition from the Principles and Practice of Sleep Medicine. It defines sleep as a reversible behavioral state of perceptual disengagement from and unresponsiveness to the environment. And I think almost every word in this definition is pretty much crucial to defining sleep. First of all, it's reversible. That's very, very important. If you're asleep, you have to be able to get up if there's an emergency or a danger present. And this is one of the ways we distinguish sleep from other states of reduced consciousness or unconsciousness, is that it should be easily reversible. It involves a perceptual disengagement from the environment, reduced disengagement, not eliminated disengagement, perceptual engagement from the environment. When you're asleep, most of your environment you're unable to perceive, but many of us have been in situations where we have had dreams about things that are in our environment, which suggests to us that the disengagement from the environment isn't absolute, but it's dramatically reduced. And the responsiveness to the environment is dramatically reduced. We usually measure that with response thresholds. For those of us who are awake now, if I whisper your name or gently tap you, you are likely to respond. If you're in a sleeping state and I do the same, you may not be as quick to respond. And response thresholds is another, not only way that we define sleep, but a tool that we use to measure a variety of characteristics about sleep. Our sleep cycle usually consists of, for humans and most mammals, 2 stages of sleep. non-rapid eye movement sleep, and rapid eye movement sleep. It's important that I define these now because in future slides I'm going to be talking about these two states. Non-rapid eye movement sleep is the stage of sleep that dominates the majority of our sleep in the nighttime. Non-rapid eye movement sleep is best characterized by lack of eye movements during sleep, sustained muscle tone. If you're in non-rapid eye movement sleep, your muscles are in a sustained tone, so you can maintain a position and even potentially move around. And no dreaming. So for most non-rapid eye movement sleep, there's no dreaming involved. Rapid eye movement sleep is a very important stage of sleep, but it's the stage of sleep that comprises the minority of the sleep during the night. It's defined by rapid oscillating eye movements under your closed eyelids. This is the state in which the majority of your dreaming occurs, and this state is also defined by reduced or complete elimination of muscle tone, so you don't act out your dreams. So even though we understand the two stages of sleep and we know that sleep is important, it has been difficult for us to figure out exactly how it works. So in order to do that, we typically subscribe or design our experiments around the two-process model, first proposed by Alexander Borbet in 1982. If you've seen any experimental sleep lectures, you may have seen some version of this model presented. It's not unusual in lectures like this one. And the two-process model in 1982, Borbet proposed that sleep was regulated, generally speaking, by two processes. The first are circadian rhythms. Those are your daily rhythms of behavior and physiology that are driven by circadian clocks in your brain and in your body. They're generated by these clocks, and they're coordinated by a master clock in the suprachiasmatic nucleus, which is in the hypothalamus of the brain. The second process is the sleep homeostatic process, and anyone that remembers the physiology learned about homeostasis of a variety of different mechanisms. Sleep works the same way. Homeostatic mechanism is a mechanism that actively defends a set point, whether it's respiration or whether it's heart rate or whether it's a variety of other different behaviors or physiological processes. There's an ideal set point that if you move too far away in either direction from, your body will actually activate processes and expend energy to try to move you back toward that point. Sleep homeostasis, in theory, works the exact same way. There's a certain amount of sleep you should be getting every night or every day. And if you get significantly less or significantly more than that amount of sleep that you need, your body will expend energy and make adjustments to bring you back to the daily amount of sleep you need. Borbet defined the circadian process as process C, and the sleep homeostatic process as process S. And we use this model to better understand sleep and also to design our experiments. I'm interested in the genetics of sleep, so I tend to use this model. According to Borbet, these two processes interact with one another to generate your 24-hour sleep-wake cycles. The homeostatic process, even though it's a real homeostat, is normally considered when we're talking about sleep loss. And in this case, when I talk about sleep homeostasis throughout most of this presentation, I'm going to be talking about the ability to recover from insufficient sleep or the ability to recover from too little sleep. A traditional homeostatic sleep rebound, when you don't get enough sleep, is, again, if you go through one night, you don't have sufficient amounts of sleep, at your next sleep opportunity, you will sleep more deeply, and you'll usually sleep longer for a longer duration. There are other components of the homeostatic response also that I'll get into later in this talk. It's a coordinated physiological process. Sleep pressure is proportional to the duration of prior wakefulness. So the longer you're awake, the deeper and longer you will be asleep at your subsequent sleep opportunity. Sleep pressure is sleep pressure. You may have heard it referred to as sleep drive, sleep debt, sleep propensity. There's a variety of names for it. Here I'm calling it sleep pressure. That's all process S that was defined by Alexander Borbet in 1982. We express it in our EEG or electroencephalogram as the power of the delta waves in your EEG, and I'll show you some figures of that in later slides. EEG delta power is also known as slow wave activity. That's the power of those delta waves or slow waves, which are the lower frequency slow waves in an EEG between 0.5 and 4 hertz. And the neurobiological and genetic substrates for sleep pressure are unknown. I have this pipe emitting water or liquid as because I like to use this metaphor for how sleep pressure works for those that are having a difficult time conceptualize it. Without having an actual substance that we assign process S, I like to think of sleep pressure as pressure in a pipe. If that pressure is closed, you're awake. And as you're awake and that pipe is closed, as the pipe is closed as you're awake, the pressure builds in that pipe. The longer that pipe is closed, the more pressure will build in that pipe. When you go to sleep, the pipe opens and it relieves that pressure. The liquid is able to flow out of the pipe and the pressure is relieved. When you wake up in the morning, that pipe is closed again and that pressure builds. Now, if you think of sleep pressure and as a pipe, two things come to mind. What is it that's opening and closing that pipe? You're getting up in the morning, that pipe closes, the pressure builds. You're going to sleep at night, that pipe opens. What is the circadian clock that opens and closes that pipe? The circadian clock is responsible for timing your preferred times of waking up in the morning and going to sleep at night. So if this pipe is responsible for your pressure, you can imagine a timer or a clock attached to this pipe that closes it in the morning and opens it at night. Now, if you close the pipe and stay awake and you don't sleep, the pressure continues to build in that pipe, the pipe will likely burst, and that's exactly what happens if you don't sleep in life. It can harm your health, it enhances your morbidity, it decreases your health, and ultimately can have lethal consequences. So this is one limb of Bourbe's model. The other limb of Bourbe's model are those clocks, the clocks that open and close the pipe. Now, we appreciate that the clocks are responsible for timing the times of day you wake up, the times of day you go to sleep, the times of day you're going to feel more alert, and the times of day you're going to feel more sleepy. But another underappreciated role of the clock is to consolidate your sleep-wake cycles. Most of us, on a given day, for the most part, we tend to be awake in the daytime and asleep at nighttime. Ideally, you want to spend about 16 hours awake and normally around the day, 8 hours of sleep at night, and our circadian clock is responsible for consolidating those. And so it just doesn't only time your sleep, but if you don't have a circadian clock, you're going to be waking up and going to sleep sporadically throughout the day, and that's been shown experimentally in a variety of models in which the circadian timing is not working. The circadian clock in the body is a real clock. It has an input mechanism, the light-dark cycle, which entrains it or synchronizes it. It has a core timing mechanism. In this figure, those are these gears. As I will go on to show you in a few slides, they are the genes that regulate circadian timing. And it has an output mechanism. In this figure, it's the hands that we tell time. For us, it could be our sleep-wake cycle. It could be our activity cycle. It could be a variety of different physiological and behavioral processes that show 24-hour rhythms. These circadian clocks exist in the majority of cells throughout your body. The master clock is in the brain, but your skeletal muscle, your liver, your lungs, your blood vessels, your heart, all of those cells and all of those organs have actual real clocks that can be set, that tell accurate time, and that reflect that time through some process. And this circadian timing mechanism is one of the focuses of my research. I'm very interested not in just the genetics of sleep, but in manipulating the circadian clock, genetics of the clock, to better understand how we sleep and sleep homeostasis. In this figure by John Webb and Ying-Wei Fu, I put this up here to show the thousands of genes and dozens of pathways, in this case, grouped into different regulatory mechanisms or structures that are direct regulators of sleep. So we know there are a variety of genes that can be grouped under transcription factors, neurotransmitter receptors, kinases, neuropeptides, and et cetera, that not only influence sleep, but are directly responsible for encoding some variety of sleep. But believe it or not, over about the past couple of decades, the story of the genetics of sleep has really been the story of the genetics of circadian clocks. This is a slide provided by Dr. Fred Turek at Northwestern University, which I enjoy using. Borbet proposed that sleep is regulated by an interaction between the circadian timing system and sleep homeostasis. In my lab, we tend to use what we know about the circadian clock to answer questions about sleep homeostasis. And over the past 20 years, we've discovered a lot about the circadian clock. In the 70s, Seymour Benzer and Ron Konopka discovered the first genetic mutation that underlied circadian rhythms in the fruit fly. After that, Jeffrey Hall, Michael Rosbash and Michael Young, Jeff Hall and Mike Rosbash at Brandeis, Michael Young at Rockefeller, discovered the first circadian clock gene, which they call the period mutant, and they discovered the timeless gene also. That was the first indication that circadian clocks were indeed regulated by genes. They discovered the first circadian timing genes. And then in 1996, Joe Takahashi, Fred Turk, and Martha Vita Turner discovered the first mammalian gene, which they called the clock gene, because when they caused a mutation in this gene, the animal failed to show circadian rhythms of circadian locomotor rhythms. It was a mouse, and they had the mouse on a running wheel, and when they induced a mutation in this one gene, the clock gene, the mouse did not show daily rhythms of locomotor activity. So they named that gene circadian locomotor output cycles kaput for the clock gene. In 1996, the first mammalian clock gene cloned. After that, there were a domino effect that led to several clock genes. And since then, a large amount of our understanding of the genetics of sleep and sleep quality has been driven by our understanding of clock genes. This cartoon shows a description of how that molecular genetic timing mechanism in the circadian clock actually works. I'm not going to belabor you. I can point out there's the clock gene again, circadian locomotor output cycles kaput, the BMO1 gene, the period genes that were discovered by the individuals I mentioned earlier, who actually won the Nobel Prize in 2017 for physiology and medicine for discovering these genes. So these genes were a major discovery, led to a Nobel Prize. Again, Jeff Hall, Mike Rosbash, and Michael Young. These are the cryptochrome genes. And without belaboring exactly how this mechanism works within every cell that has a circadian clock, you can think of these as the core timing mechanism for circadian rhythms. These are the gears on any clock that's run by gears, and not a whole lot of those anymore. It's the electrical timing mechanism in any clock that keeps time. This molecular feedback loop is driven by the transcription and translation, the expression of these genes in a cyclical manner, and the time constant of this feedback loop of genetic and molecular interactions defines the period of your circadian clock, which is usually very close to 24 hours. It's entrained to 24 hours by the daily light-dark cycle. So over the last 20 or so years, we've learned a lot about how the circadian clock works, and we've learned a lot about the genes and molecules that underlie the circadian clock. But what we've been surprised by, particularly over about the last 15 years, is that these circadian clock mutations in a variety of species have effects on sleep and wakefulness that we don't necessarily associate with timing, right? Borbet's model suggested that the circadian clock timed and consolidated sleep-wake states, and that sleep homeostasis was responsible for determining things like the total amount of sleep we need a night and the ability to recover from sleep loss. However, over the past 20 or so years, we've discovered that these circadian clock genes may do more than just time or consolidate sleep. This is a study in 2007 done in the lab of Dirk-Jan Dijk that found that the circadian timing gene period 3, in humans, there's a polymorphism of that gene. That means there's a genetic variety, an allele of that gene, that if expressed has an effect on both sleep amount in REM and non-REM sleep, slow wave activity in REM and non-REM sleep, and the ability to recover from sleep deprivation, here shown in recovery non-REM sleep and baseline non-REM sleep. REM sleep here and slow wave activity here. The ability to recover from sleep loss and the total daily amount of sleep can be influenced by different polymorphisms of this one gene. If you have one allele, you're likely to sleep a certain amount of the day. And if you have another allele, your sleep amount is likely to be different. In 2009, in the lab of Yingwei Fu at the University of California, San Francisco, she found another one of these circadian clock genes that are normally associated with circadian timing, that there is an allele or polymorphism of this gene that encodes sleep length in humans. This is another gene that we typically associate with circadian timing, but appears to be responsible for determining the amount of time that you're going to be sleeping in a night. And there's a third gene, these two I showed in previous slides. This clock gene, again, there's that clock gene discovered by Martha Vita Turner, Fred Turek, and Joe Takahashi in '96. This lab, led by Theo Ronenberg, found that various variants or genetic alleles of clock were associated with sleep duration in two independent populations. So here we kind of have a twist on what we typically saw as Bourbe's classic model of sleep. We see that these specific genes, if you think of the gears of the clock, may not just time sleep-wake states, but may be responsible for things we normally associate with sleep homeostasis. In my lab, we examine sleep in the mouse model. For a variety of reasons, the mouse is a very effective mammalian model of behavior, and it's also one of the most effective genetic models of behavior. Mice are most commonly used to try to better understand the genetic origins, genetic factors that drive behavior and physiology. But before we started our experiments that I'm going to show you today, I wanted to give you a little background of some of the things that have already been done in mice to suggest that circadian timing and circadian genes may be responsible for regulating sleep-wake states, and particularly sleep homeostasis. In this 2000 paper, Eric Naylor and Fred Turek showed that this mutation of this clock gene that I mentioned earlier alters sleep and wake amount. This gene shows the wild-type mouse, the double mutant clock mouse, and the clock heterozygote over 24 hours of sleep EEG recording and a 12-hour light, 12-hour dark cycle. This is the sleep amount during the light phase, in which the mice are nocturnal. Sorry, during the daytime, they're normally sleeping. During the nighttime, they're normally active. This shows the sleep amount during the dark phase. This graph here on the right is the one I would like to draw your attention to. This shows percentage of recording of EEG in wake, non-REM, and REM sleep over 24 hours of recording. So just a typical 24-hour day. And as you can see, the mouse that shows the clock mutation is awake more than the wild-type mouse. And that additional wakefulness comes at the expense of primarily non-REM sleep. This mouse shows a less non-REM sleep over 24 hours than the wild type. So this is, as we've seen in humans, in a mouse model, a mutation of a circadian gene is responsible for having less sleep. This is another gene. This is one that my lab has taken particular interest in, the BMAL1 gene, which is another circadian clock gene. These graphs on the left show 24 hours of continuous EEG recording in 12 hours of light, 12 hours of dark, over 24 hours for wild-type mice that are heterozygote for the BMAL1 deletion mutation, a knockout, and mice that are homozygous for the BMAL1 deletion mutation. This is eliminating BMAL1, both alleles of BMAL1 in a mouse. These graphs on the right are the graphs I would like to draw your attention to. This one shows total sleep time over 24 hours. This one shows total non-REM sleep. As I said earlier, that's the majority of sleep in humans and mice. And this one shows REM sleep over 24 hours. And when you knocked out, when Aaron Leposki and Fred Turek knocked out BMAL1, they found the opposite of what they saw in the clock mutant mouse. This mouse had more total sleep, more non-REM sleep, and more REM sleep over 24 hours of recording. And the majority, if not all of that effect, was seen during the dark phase when mice are usually active. Mice are not absolutely biphasic like humans. Mice mostly sleep in the day and mostly are awake at night, but they do sleep a little at night and they are awake a little in the day. And this mutation, the BMAL1 knockout mouse was arrhythmic, this flat line here, did not show rhythms of sleep and wakefulness across 24 hours. And as a result, the mouse had more sleep, and the majority of that sleep occurred during the dark phase, again, which is when the mouse is normally awake. So we have two circadian mutations, one causing increased wakefulness, one causing increased sleep, both showing that circadian clock genes, the genes that are responsible for timing our daily behaviors and rhythms of behavior and physiology, may actually contribute to sleep amount. In my lab, we were really interested in that BMAL1 knockout that Aaron Lepofsky and Fred Turk published. BMAL1 is a critical gene. It's both necessary and sufficient for circadian timing, and it's the only gene that if you eliminate it, you eliminate circadian rhythms. The other genes are important for circadian rhythms, but if you get rid of them, there are compensatory mechanisms. There's a bit of redundancy built into those genes. BMAL1 obliterates the circadian clock. As I showed you in the previous slide, Lepofsky and Turek showed that BMAL1 knockout, that means you delete both alleles, increase baseline sleep amount at the expense of wakefulness, they increase baseline slow wave activity, and they attenuate the ability to recover from sleep loss. This decrease in slow wave activity is important. I told you earlier about sleep pressure in Bourbe's model, that's process S. Our best way of measuring sleep pressure is by affixing EEG to humans or to mice and looking at the slow wave activity during non-REM sleep. Non-REM slow wave activity is the power in microvolts of your EEG bandwidth between 0.5 and 4 hertz. The majority of non-REM sleep is dominated by these slow waves of your EEG, and the power, the electrical power, we're electrical beings. Our EEGs are measuring the brain waves, the oscillation of brain waves during all of your states, and we use the electrical power of your EEG during non-REM sleep to measure the depth of your sleep. And that is our best and most reliable measure of sleep homeostasis. We call that slow wave activity. We also call it non-REM delta power. As you can imagine, when I talk about sleep pressure and sleep drive, my field has a hard time agreeing on names for things. So we can use interchangeably non-REM delta power or non-REM slow wave activity is measured in slow wave sleep. And Dr. Lepofsky found that this mutation increased baseline slow wave activity and attenuates the ability to recover from sleep loss. So what do? We got a mouse model from Joe Takahashi and Aaron McDermott, which knocked out BMAL1, but rescued BMAL1 specifically in the skeletal muscle and in the brain. We know that there are clocks throughout the body, and we knew that BMAL1 was enriched in skeletal muscle and the brain. So we decided to ask the question, what tissue was driving most of these phenotypes that we were seeing? You know, we kind of took for granted that it would be the brain at the time, but we decided to do these experiments anyway, just because that's what we do. We try to understand how things work, and we were trying to figure out the genetic nature of sleep. So without belaboring how this was done, we got these models kindly donated to us from Joe Takahashi, who at the time was at Northwestern University, and they rescued BMAL1 function in the skeletal muscle of mice that did not express BMAL1. So you can imagine a mouse that does not have this gene, and we express this gene in the skeletal muscle molecularly, and then we take another mouse that does not express the gene, and we molecularly express the gene in the brain. And we took those mice, and we surgically implanted them with electrodes for EEG and EMG recording of sleep-wake states. And we maintained them in a 24-hour light and a 24-hour dark, 12-hour lights, 12-hour dark, 24-hour recording light-dark cycle, much in the vein of the data that I showed you earlier. Our first surprising was pretty finding. These experiments were done by Chris Allen at Morehouse School of Medicine when I was still a faculty member at Morehouse School of Medicine. He's currently an associate professor at Morehouse School of Medicine in Atlanta. And the first finding from these studies was that rescuing this gene, BMAL1 in the brain, did not restore our sleep amounts. This graph shows 24 hours of non-REM sleep. This one shows REM sleep, and this one shows non-REM slow wave activity. All three over 24 hours of recording. Again, non-REM slow wave activity is our best measure of sleep pressure. But I would like to draw your attention to the graph on the left because that was our most surprising result. We looked at sleep in wild-type healthy mice. We looked at sleep in mice that did not have BMAL1 anywhere. And we looked at sleep in mice that rescued BMAL1 in the brain. And we found that rescuing BMAL1 in the brain did not rescue the sleep phenotype. The mice that expressed BMAL1 in the brain slept just like mice that had no BMAL1 and significantly more than mice that were healthy, wild-type mice, BMAL1 throughout. So this gave us suggested to us that the effect of the mutation wasn't being driven by sleep regulatory mechanisms in the brain. We then looked at the mice that rescued BMAL1 in the skeletal muscle, and to our surprise, those mice slept like wild types. Again, it's the graph on the left I would like to draw your attention to. Rescuing BMAL1 in the skeletal muscle reduced the sleep amount significantly lower than in the knockout mice. These mice slept the same as wild-type mice. So that was a eureka finding to us, that a gene expression in the skeletal muscle, a clock gene no less, expressed in the skeletal muscle, could affect total sleep amount, since we know that sleep is driven by processes in the brain. But we didn't know if it was directly driven by sleep regulatory mechanisms or if it was an ancillary effect of the mutation. These mice have a variety of phenotypes. If you express a mutation in skeletal muscle, it could cause a variety of what we call ancillary or pleiotropic effects. Mouse could be in pain, mouse could be uncomfortable, it could be a variety of things to influence sleep. How do we know that the mutation that we induced is directly acting on sleep regulatory processes? Well, one way to do that is by a classic tool we use called a homeostatic challenge. And it sounds fancy, homeostatic challenge, but all it does is apply forced wakefulness to a mouse for 6 hours, sleep deprivation, 6 hours of sleep deprivation, and allow that mouse to recover. Remember, sleep homeostasis is a classic, reliable response to sleep deprivation. And in a healthy mouse or in a healthy human, it's very predictable. 6 hours of sleep deprivation, forced wakefulness, done by a gentle handling technique. The gentle handling technique requires just standing in front of a mouse cage and tapping a cage, tapping the cage. And if the mouse gets too sleepy, you prod the mice. To date, it's the only way we know of ensuring absolute sleep loss. We've tried a variety of automated mechanisms, and mice are really smart. No matter how sophisticated the mechanism, the mouse usually finds a way to sleep. So in order to keep a mouse awake, you have to be really good at convincing undergraduates to come into your lab, stand in front of cages, and tap those cages. And they have to do them in 1 hour shifts for 6 hours, or else another undergraduate will have to come in and keep them awake. So we have undergraduates that do gentle handling, tap on cages for 6 hours, and then we allow the mice to recover. A classic rebound response. This is just data for sleep rebound. This is what baseline sleep, non-REM sleep, and baseline REM sleep looks like in a mouse. If you sleep deprive a mouse for 6 hours, it's not a robust amount of sleep deprivation, but it is sufficient for us to get a classic rebound response. And when we do that, you usually get an increase of non-REM and REM sleep, usually in the second 6 hours of the light phase, but it can often extend into the dark phase of a 24-hour light-dark cycle. You get, like I said earlier with sleep homeostasis, you'll get more sleep and you'll get higher intensity sleep. And what does deeper sleep look like? Well, deeper sleep looks like non-REM relative delta power, or we call it slow wave activity. Slow wave activity we measure during non-REM sleep. So in the white dots, in the white diamonds, the sleep-deprived mice, we don't measure slow wave activity during the first 6 hours because the mice don't have any non-REM sleep in the first 6 hours. But during recovery, during the non-REM sleep bounces, you can see their non-REM delta power is significantly higher than in the baseline mice. This is a classic recovery response for a 6 hour homeostatic challenge. It drives up non-REM delta power, again, our best measure of sleep homeostasis, and non-REM delta power reflects sleep pressure. So if you sleep deprive a mouse for 6 hours, you get this significant increase in sleep pressure. And again, it takes the mice all the way up until the dark phase, until that sleep pressure is dissipated and becomes similar to baseline mice. So 6-hour sleep deprivation challenge is not only very reliable at testing the sleep homeostat, we also know it's a brain-driven process. We know the sleep regulatory mechanisms in the brain are responsible for sleep homeostasis. So in our brain-rescued and muscle-rescued mice, we did that. We did a 6-hour sleep deprivation. We looked at recovery sleep amount as a percentage over the same amount that they slept over baseline. And in our brain-rescued mice, they recovered like knockouts. The rescue of BMAL1 in the brain did not restore sleep amount. In our muscle-rescued mice, we rescued BMAL1 in the skeletal muscle, and these mice, it completely restored their sleep response. This is, again, slow-wave activity, our best measure of sleep pressure, again, recovery over corresponding baseline, and rescuing BMAL1 in the skeletal muscle restored their homeostatic sleep response, suggesting to us that the muscle mutation was indeed either talking to or acting on sleep regulatory mechanisms in the brain. Here we have what are called spectral curves. Along the horizontal axis is the total frequency of the EEG up to 30 hertz, and along the vertical axis is the power in microvolts, microvolts squared, of that EEG. Again, during non-REM sleep, the majority of that EEG is going to be with slow waves between 0.5 and 4 hertz, as shaded in yellow on these graphs. And that's where you're going to see the peak of EEG during non-REM sleep, again, reflecting sleep homeostasis. In wild types, in our mice, we rescued BMAL1 in the skeletal muscle. Their non-REM slow wave activity during recovery was not different from wild types. Robust, which you would expect after sleep deprivation. Our brain-rescued mice were not different from knockouts. The spectral power of the EEG bandwidth in the slow waves was reduced. I would like to use the word impaired, similar to knockouts, suggesting to us that rescuing this clock gene in the brain was not sufficient to restore these sleep phenotypes, rescuing the gene in the skeletal muscle. But what did we do? We showed that BMAL1 in skeletal muscle is sufficient for healthy sleep. Isn't necessary. We decided to be rigorous. We see it's sufficient. Is it necessary? We rescued BMAL1 in the skeletal muscle of mice that didn't have BMAL1. The next thing we did was knocked it out exclusively in skeletal muscle of healthy mice. We did the reverse. We took healthy mice. Karen Esser actually did this at the University of Florida. She donated these mice to us. That's knocked out BMAL1 in the skeletal muscle, and this knockout was could be turned on and off by tamoxifen. So this is one of the reasons the mice are such a valuable genetic tool for these studies, because you can do cool things like this in a mouse that you can't do in other species. You can knock out a gene in the skeletal muscle, and you can turn it on and off by putting tamoxifen in the drinking water, kind of like a switch. Turn BMAL1 on in the skeletal muscle, turn it off in the skeletal muscle. This work was done by Alison Brager, who at the time was a postdoc in my lab when I was still at Morehouse School of Medicine. And it's the graph on the left I would like to draw your attention to, because that shows the effect that we saw when we knocked out BMAL1 exclusively in the skeletal muscle, only tissue that doesn't have BMAL1. It increased non-REM sleep just like a whole body knockout would. The skeletal muscle knockout was sufficient to make the mouse sleep like a whole body knockout, suggesting to us that BMAL1 is indeed critical for sleep, and that that knocking it out in the skeletal muscle was allowing it, having influence on sleep regulatory mechanisms in the brain. So we rescued BMAL1 in the skeletal muscle of knockouts. We restored sleep phenotypes. We knocked it out exclusively in skeletal muscle, and we impaired sleep. But there's still significant questions, right? As I mentioned earlier, if you knock out BMAL1, you break the clock. The clock doesn't work without BMAL1. So are our phenotypes driven because we don't have BMAL1 or because we don't have a clock? Is it the gene or is it the mechanism? And we were scratching our heads at the best way to get at that question until we realized that the littermates of our rescue mice actually express BMAL1 in the skeletal muscle and brain of wild types, an exogenous BMAL1. And when we do that, that overexpresses BMAL1 in the skeletal muscle, and it overexpresses BMAL1 in the brain. We express that transgene, we express that artificial gene on a healthy mouse, and we ramp up BMAL1 levels in the skeletal muscle and in the brain. When we do that, we change the amount of BMAL1, right? You alter the amount of BMAL1 gene, RNA, and protein, but the clock still works fine. Ramping up BMAL1 has no deleterious effects on the circadian clock. The clock still functions, the clock's still there, but we get to see if changing levels of BMAL1 have any effects on sleep. And for time's sake, I'm only going to show you what we found in the overexpression model, because that was our biggest finding. Here we have 72 hours, 3 24-hour days of a 12-hour light, 12-hour dark, 12-hour light, 12-hour dark. This is a healthy mouse, a wild-type mouse and a BMAL muscle overexpression mouse, baseline ad libitum sleep-wake conditions. Here is sleep restriction. We had to use sleep restriction because I am pretty good at convincing undergraduates to tap cages for 6 hours. Not good at convincing undergraduates to tap cages for 24 hours. So here we had to use an automated sleep deprivation tool, a slowly rotating wheel. The wheel moves around at 1 RPM. The mice have to be active throughout the 24 hours. And this is non-REM sleep. As you can see, both our wild types and our BMAL1 overexpression are able to sleep. Like I said, these are smart guys. They have what are called micro sleeps. These wheels, if you can imagine, a wheel slowly moving and the mouse having to stay active. And when the mouse gets really tired, he's just going to try to sleep, and then he's not going to be able to. He figures out that if he runs up one part of the wheel, he can ride it around for a few seconds until he gets to a point that he can't ride it again. He can wake back up and do the same thing over again. You can watch these mice do it. It's pretty incredible. These are called micro sleeps, and if you get enough of them, as the mice get tired, they will have significant amounts of sleep. Doesn't do much to reduce their sleep pressure, because really short bouts of sleep, highly fragmented sleep, doesn't do well to reduce sleep pressure. But they still get non-REM sleep, and then during the recovery, after we had them in the slowly rotating wheel, we allowed them to recover in 24 hours. The first thing that jumped out at us was the mice that overexpressed BMAL1 appear to be sleeping less than the wild-type mice. And when we quantified that here, we did find out that in the wheels, the mice that overexpress BMAL1 are indeed awake more, sleeping less, suggesting to us that overexpressing BMAL1 in the skeletal muscle makes the mice protective against sleep loss, maybe. They're sleeping less, they're easier to keep awake in these wheels than wild-type mice. Something about overexpressing this gene in the skeletal muscle is making these mice a little bit more vigilant. If you look at recovery, looks like there's no difference in sleep amount during recovery. However, you gotta remember that the wild-type mice had more sleep, significantly more sleep during sleep deprivation than the BMAL mice. So if you look at non-REM sleep amount and you adjust for the differences in the amount of sleep lost, you measure non-REM sleep amount as sleep gained during recovery divided by sleep lost during sleep deprivation, you see that the mice that overexpressed BMAL1 in the skeletal muscle only recovered half the sleep as wild-type mice, suggesting that overexpressing this gene in the skeletal muscle was sufficient seem to be protective against sleep loss. This graph shows that the non-REM delta power throughout the experiment was lower in mice that overexpressed BMAL1 in skeletal muscle, suggesting lower non-REM delta power suggests to us that these mice are indeed less sleepy. Delta power, again, is our best measure of sleep pressure. If you have a high delta power, you have a higher sleep pressure. You have a low delta power, you have a lower sleep pressure. But this is the one that jumped out at us because we measured slow wave activity during waking. Now, normally you can't get any slow wave activity during waking because when you're awake, most of your waves are fast. You have sigma waves, you have beta waves. Your brain's doing a whole lot of things when you're awake. Different parts of your brain are firing different frequencies. Your EEG is fast. You don't have a lot of slow waves. But if you sleep deprive a mouse or a human, you begin to get more slow waves in your waking EEG. The sleepier you guys get, hopefully you're not sleepy now, but the sleepier you guys get, the more, if I have an EEG on you, the more slow waves will be in your waking EEG. And in this case, we found that mice that overexpressed BMR1 in the skeletal muscle had more slow waves in their waking EEG than wild-type littermates, substantially more slow waves, suggesting to us that not only were these mice awake more, these mice were able to incorporate more slow waves into their waking EEGs, almost like the brain can stay awake but kind of be asleep at the same time, suggesting to us that these mice overexpressing BMR1 in the skeletal muscle was making these mice protective against sleep loss. So what I've showed you so far, this gene that's associated with circadian timing, not only can affect sleep, but its expression in skeletal muscle is important in its ability to regulate sleep. This gene, BMR1, if you rescue it in the skeletal muscle of knockout, it restores sleep phenotypes. If you knock it out in skeletal muscle, it reduces recovery from sleep loss and increases slow wave activity. These are components of sleep homeostasis, suggesting to us that expression in the circadian clock is having effects on the homeostatic mechanism. BMR1 overexpression in skeletal muscle reduces recovery from sleep loss, reduces slow wave activity, and appears to make mice resistant to sleep loss. And it is necessary for the proper functioning of sleep regulatory mechanisms. But what does resistance to sleep loss look like? Are they resistant to sleep loss? Well, we decided to do a classic memory test. This is a novel object recognition test. Without belaboring you too much, we usually allow the mice to habituate to an arena. We place 3 objects in the arena on the training day. Then the mice becomes familiar with those 3 objects. Then between the training day and the testing day, the mice are either sleep replete. This is not sleep restricted, this is actually sleep replete. Sleep replete means, SR in this case, means the mice have sufficient sleep. They're sleeping ad libitum. So they're either 12 hours that they're sleep replete, or we have 12 hours of sleep deprivation. And then we give them, we put a new object in the arena, and the mice usually, if their memory is preserved, they will spend more time around the novel object because they'll recognize it as new. They'll remember these other 2 objects. So we did this in our wild types and our mice that overexpress BMR1 in the skeletal muscle. This is sleep replete. These are mice that have not been sleep deprived. These are sleep deprived mice. And when we look at our wild type, B6 is the strain of the mouse, we see that the sleep-deprived mice are showing a classic response to sleep deprivation. They're spending less time around the novel object, more time around the familiar objects because there's a memory impairment. This is a very reliable test of memory. However, the mice that overexpress BMR1 in the skeletal muscle are spending more time around the novel objects, suggesting that their memory is indeed protected from sleep deprivation and providing support for our hypothesis that overexpressing BMR1 in the skeletal muscle does indeed protect memory against impairments of sleep deprivation. These are the same data on the graph on the left and the right for sleep replete and sleep deprived. This is each mouse, first before they were sleep deprived and after they were sleep deprived. There's only one BMR1 overexpression that did worse than the sleep deprived condition. All of the rest not only preserved their memory, but for some reason did better, and we're still not able to figure out why that is. And the wild-type mice performed as expected. Then we actually examined BMR1 levels in the skeletal muscle of our wild types and our knockouts, and we graphed them, the levels of BMR1 expression correlated against their performance in the novel object recognition test, and we found a significant effect of BMR1 expression in the muscle. The more BMR1 they had expressed in the skeletal muscle, the better they did in the novel object test after sleep deprivation, suggesting to us that expressing this gene in skeletal muscle is having beneficial effects on memory after sleep deprivation. And the conclusion of this is that overexpression of BMR1 in skeletal muscle confers resistance to the negative effects of sleep loss and memory. Since we have a little time left, I'm going to jump ahead and show you some of what we found in the skeletal muscle of BMR1 muscle overexpression mice. First, overexpressing this gene in the skeletal muscle does have effects on mean cross-sectional area of the skeletal muscle, but does not have significant effects on the total myofiber number of skeletal muscle. So overexpressing BMAL1 in the skeletal muscle, we're really interested in what it's doing in the muscle and in the brain. But we're finding out really interesting things. It's in the muscle, it's increasing cross-sectional area. There's a lower energy expenditure for this active BMAL1 is the actual transgene we use to overexpress BMAL1 in the skeletal muscle. So active BMAL1 are mice that have higher levels of BMAL1 in the skeletal muscle. We put them on little treadmills and an oximax system that can measure gas exchange during a workout. And that measurement of gas exchange indicates energy expenditure. And we found that mice that overexpress BMAL1 in the skeletal muscle have lower energy expenditure than wild-type mice. We found that sleep deprivation decreases BMAL1 levels in the skeletal muscle. I'm jumping through this relatively quickly. And we found that the BMAL1 signaling cascade in skeletal muscle is altered. And this is very important because BMAL1 is higher in mice that overexpress BMAL1. We found that BMAL1 is a transcription factor, which means it can activate and deactivate other genes. One of the genes that BMAL1 acts on is PGC-1α. We found that that was also increased in skeletal muscle. But we have a little bit of hints as to what may be the signaling mechanism to sleep regulatory systems in the brain. That is because brain-derived neurotropic factor is a cytokine. It's secreted from the muscle, much like a hormone is secreted from glands. And BDNF acts on mechanisms in the brain. We know that BDNF that's in circulation can act on the brain to influence a variety of different behavioral states. And in mice that overexpress BMAL1, we're getting a reduction in BDNF in the skeletal muscle. We're still trying to determine what effect that has on the brain. So we have found that overexpression of BMAL1 in the skeletal muscle alters metabolic activity and signaling cascades in skeletal muscle. Very interested in the genetics of sleep. We want to understand the genetics of sleep homeostasis. Findings from other labs and findings from my lab suggest that genes that regulate the circadian clock may be a lot more important for sleep and the ability to recover from sleep loss than previously thought. And work from my lab suggests that those genes don't necessarily have to be expressed in the brain, that clocks in the periphery have effects on sleep regulatory mechanisms. In this case, specifically, clocks and BMAL1 in the skeletal muscle is having effect on sleep regulatory mechanisms, showing that circadian clocks throughout the body don't necessarily just regulate circadian rhythms in the body, but can feed back to the brain and regulate sleep. I would like to thank everyone who participated in the study. Most of the data I showed you today was done by Chris Aylen and Allison Brager, who were at Morehouse School of Medicine at the time. India Nichols and Alexis Tucker at UCLA did the lion's share of the unpublished data that I showed you, and our mouse models were donated to us by Karen Esser, Joe Takahashi. The energy expenditure experiments were done by Colleen Novak at Kent State University. And I'll stop there and open the floor for questions. Thank you for having me.